Viewers, you're watching Student's Diary, and today's topic is about citric acid cycle, which is also known as Krebs cycles and TCA cycle as well. Citric acid cycle was first proposed by Hans Adolf Krebs in 1937, and he has been awarded a Nobel Prize for his tremendous work. Citric acid cycle is essentially involves in oxidation of acetyl-CoA to carbon dioxide in water. We know that in a carbohydrate metabolism, especially in glycolysis process, we get the pyruvate, which is then converted into a two carbon molecule, that is acetyl-CoA, and which contain pretty much energy in it, and then it is converted into a lower energy compound, which is carbon dioxide. And all this process of citric acid cycle take place in mitochondrial matrix. If we talk about the energy process of this citric acid cycle, it is the most predominant pathway because it gives you 65 to 70% of the total ATP synthesized in our body. So that give you a pretty much a large quantity of ATP, and similarly it also utilized about two-thirds of the total oxygen consumed by body. So that is quite large amount of oxygen which is consumed for the citric acid cycle, and that also indicate that citric acid cycle is dependent on oxygen. If there is a deficiency of oxygen, citric acid cycle will not work. Similarly, this citric acid cycle also provides many intermediates that is required for synthesis of amino acid, glucose and heme. So this citric acid cycle is not a closed circle, but rather it's a kind of a roundabout where different vehicles came in and go out. Similarly, in case of biomolecule, at the time of demand, this citric acid cycle can give you certain amino acids, and that will be of course non-essential amino acid, and it also give you glucose molecule in the form of gluconeogenesis and heme and fatty acid, etc. That's why it's called as central pathway, because it's connecting almost all of the pathway directly or indirectly. So it is also known as amphibolic process because it gives you catabolic as well as anabolic reactions. So for understanding that citric acid cycle, we must know about the basics of the mitochondria, because this happen in the matrix of the mitochondria. So this is the mitochondria organelles, which is a double membrane structure. Outer membranes are smooth while inner membranes are folded enough, and that is known as crestes, while the space between it is a matrix. So that intermembrane space is filled with fluid, same is case with the matrix, and the reactions of the citric acid cycles takes place in the matrix. One thing is important to remember that the number of the mitochondria will shows about the states of the cells. If the cells are metabolically inert or less active, such as Schwann cell, it will has fewer mitochondria, while those cells which are metabolically active, such as hepatocytes and myocytes, which are cells of the muscles, it contained a larger number of the mitochondria. So similarly, athletes has cells has contain larger number of mitochondria because they are metabolically very active due to their high muscular activity. So if you look into the overall process, we know that this citric acid cycle required oxygen, and that oxygen comes to the cell through cell membrane, and similarly glucose came into the glucose through glucose transporters inside the cell. So once it is inside the cell, then certain process takes place in the cytoplasm in the form of glycolysis. We know that glycolysis could be aerobic or anaerobic, so it is not strictly dependent on oxygen. So that glucose glycolysis process will give you pyruvate, and pyruvate will be converted to acetyl CoA. Acetyl CoA will enter into the mitochondria, and in the mitochondria matrix that citric acid cycle will takes place. And from citric acid cycle, electron will be transferred by NADH to electron transport chain, where these NADH will be cached in the form of ATP. So in that whole process, certain substrate level ATPs are produced, such as in glycolysis 2 ATPs are produced, while in citric acid cycle 2 ATPs are produced. However, the NADH which are produced in the glycolysis process as well as in the citric acid cycle will be transferred to the electron transport chain, where it will be converted into ATPs. So overall there will be 32 ATPs will produced in electron transport chain. So this whole process, if we calculate all these ATP, it will give you 36 ATP. Now little about the evolution of the aerobic oxidation. We also know as that Krebs cycle. This aerobic process of oxidation or TCA cycle developed later on when the earth came into existence. The reason behind that, oxygen was not available at early terrestrial environment. So if you look into the bacteria back 3.5 billion years ago, we will find that only the glycolysis process are present. The genes of glycolysis is present in those bacteria, and because there was no such membrane bound organelles present, so mitochondria was not present in those bacteria at that time. Then with the passage of time, when we look back to 2.7 billion years ago, when oxygens were available a little bit on this earth environment, then that photosynthetic bacteria, cyanobacteria, and um, proto-blue-green algae came into existence, and this provide oxygen to the earth environment. So the oxygen of the earth start increasing day by day. And then if we come a little more uh, advanced towards the eukaryotic life, it came into existence after 1.5 billion years ago, and these eukaryotic cells or organisms have membrane bound organelles, especially its mitochondria present in it, and that with the presence of that mitochondria, that TCA cycle was more active by utilization of the oxygen, because at this time oxygen is available on the earth. So if we look into the genetic analysis of those species, the glycolysis, the genes which are responsible for the glycolysis, they are highly conserved. Means these genes for glycolysis will be present. You will find it in bacteria, which is very ancient, that is 3.5 billion years ago, and certain other bacterias, and as well as in eukaryotic cells. So that give you the evolutionary studies that the glycolysis process is very ancient process as compared to the Krebs cycle, which has been developed later on when the oxygen was available. Similarly, there are processes you will find it in every organism, irrespective of that it is aerobic or anaerobic, bacteria or eukaryotes. So these three processes are glycolysis, DNA replication, and protein synthesis. Now we will discuss about the steps of the citric acid cycle. It is consist of 8 steps, and we know that pyruvate is a 3 carbon molecules, and a single glucose molecule give you 2 pyruvate, and that pyruvate is converted into 2-acetyl CoA with the help of pyruvate dehydrogenase enzymes. Once that 2 carbon acetyl CoA combined with oxaloacetate which is a 4-carbon molecule present in the citric acid cycle. So citrate synthase enzyme will act on it, and both of these will be converted into a citrate molecule. This is the same molecules which we had discussed in glycolysis, and it has a inhibitory effect on phosphofructokinase, one enzyme which is a potent enzyme of glycolysis process. So this citrate is also used in the form of citric acid in different flavoring agents in beverage industries, and it has a lot of other uses. So that 6-carbon molecule of citrate with the help of aconitase enzyme is converted into isocitrate, and then that isocitrate will be acted upon by the isocitrate dehydrogenase, and here the decarboxylation process will take place, means one of the carbon will be removed, and when decarboxylation process takes place along with the dehydrogenase enzyme, one NADH molecules will be released. So this will be the first NADH which will be released, which will be again cached later on in electron transport chain. So with the help of this decarboxylation, that isocitrate will be converted into a 5-carbon molecule, but rather an important molecule, that is alpha ketoglutarate. If you remember, we had studied that in urea cycle, that the contribution of alpha ketoglutarate, and that alpha ketoglutarate will be further act by alpha ketoglutarate dehydrogenase complex. This is a kind of important enzyme, as we had studied as pyruvate dehydrogenase enzyme in the previous lecture. It has a similarly important role, and this will also give you another NADH molecule with, of course, with the removal of another carbon dioxide molecule. So that will result in a formation of the succinyl-CoA. Succinyl-CoA is now a 4-carbon molecule, which has been a molecule of 4-carbon in the form of oxaloacetate, which has been initiated initially and now become again a 4-carbon molecule. So that succinyl-CoA will be further acted by succinyl-CoA synthetase enzyme, and with the release of one ATP or a GTP molecule. This is a substrate level phosphorylation process. It will be converted into succinate. Succinate will be then acted by another enzyme, that is succinate dehydrogenase, and it will give you this time an FADH2 molecule that will also be again transferred to electron transport chain, where it will give you 2 ATPs, and that succinate with the help of succinate dehydrogenase will be converted into fumarate. Fumarate again will be acted by an enzyme which is a fumarase, and that fumarase will convert that fumarate into malate. Now this malate is ready to be reconverted back into oxaloacetate at the help of malate dehydrogenase enzyme. So this time around it will give you another NADH molecule. So that oxaloacetate will be regenerated, and this process of the Krebs cycle will continue with the addition of the another acetyl-CoA. So overall it will give you a 3 NADH bioutilization of a single acetyl-CoA, and one FADH2 and one substrate level ATP. But we know that a single glucose molecule will give you 2 acetyl-CoA, so this process will continue twice. So overall a single glucose molecule which will convert it into 2 acetyl-CoA will give you 6 NADH, 2 ATP and 2 FADH2 molecule. So the energetics of the citric acid cycle can be understood from this table. These are the reaction which we had discussed earlier in this cycle. These are the enzyme, as we had seen that the first enzyme is isocitrate dehydrogenase, which give us NADH, but the process will happen twice for a single glucose, so it will give you 2 NADH. But when it comes to the ATP, This is debatable when it will be transferred to electron transport chain. So a single NADH may give you 2.5 or 3 ATPs depending on which process it's going through. So we will consider right now that it gives you a 6 ATP. Similarly, alpha ketoglutarate dehydrogenase with the help of this reaction will give you again 2 NADH molecule, which is be considered as 6 ATP in electron transport chain ultimately. Similarly, succinyl co-synthase give you substrate level phosphorylation of 2 ATPs, so again 2, and succinate dehydrogenase giving you 2 FADH2 molecule. We know that certain depending again in electron transport chain whether it give you 2 ATP or 1.5 ATP, but however here we will consider about the 4 ATPs. And last but not the least, the malate dehydrogenase. This enzyme give you 2 NADH molecule as well. So overall, if we consider the number of ATP generated by citric acid cycle is 24 ATPs. Now we will look into the regulation of the citric acid cycle. This citric acid cycle is strictly regulated, especially these three enzymes are involved which has regulated the citrate synthase, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase complex. Citrate synthase is negatively regulated, or we can say is inhibited by lot production of ATP, NADH, acetyl-CoA and succinyl-CoA. When the concentration of ATP, NADH will be high, it will has an inhibitory effect on the citrate synthase. So the process of the citric acid cycle will be halted over here and it will be not converted into citrate. Similarly, if the adenine diphosphate levels are high, it will promote the isocitrate dehydrogenase to carry out the process of the citric acid cycle by converting isocitrate to alpha ketoglutarate. So low level of ATP will enhance this process. However, again ATP and NADH will have an inhibitory effect on isocitrate dehydrogenase. And third, the NADH level, high level of NADH will also have an inhibitory effect on alpha ketoglutarate, and the succinyl-CoA, which is the product of the alpha ketoglutarate with the help of alpha ketoglutarate dehydrogenase, it will have the product inhibitory effect on the alpha ketoglutarate. So high amount of succinyl-CoA will have an inhibitory effect on alpha ketoglutarate dehydrogenase complex, as well as it will has an inhibitory effect on citrate synthase as well. Another important thing to remember that low level of ATP overall in the cell will result in that no more NADH which has been produced through the citric acid cycle or glycolysis may not be converted into ATP in electron transport chain, because adenine diphosphate will not be available to be phosphorylated into ATP. So that ATP that will result in that if there will be low ADP means the NADH level will be high and it will not be converted into ATP. So if NADH level will be high, no NAD or FAD will be available to be converted into NADH further. So overall, low ADP will have an inhibitory effect on overall citric acid cycle. Now come to the amphibolic nature of the citric acid cycle. We already mentioned that citric acid cycle is not a closed circle cycle, but it has some synthetic as well as degradative effect. It has an anabolic as well as catabolic pathway, and we know that in citric acid cycle it can give us a non-essential amino acid from oxaloacetate, which is converted into aspartate with transamination reaction, and then that aspartate is converted into non-essential amino acid and also give you purine and pyrimidine at the time of their demand. Similarly, alpha ketoglutarate, again with a transamination reaction, if the body demand, can be converted into glutamate, and which is a non-essential amino acids, and it can also be converted into purine. Alpha ketoglutarate has been, we had discussed in alanine cycle, so it will also be utilized over there in urea cycle as well. That oxaloacetate can also be converted with pyruvate carboxylase enzyme into a pyruvate. So once pyruvate is a form that can be converted into glucose by gluconeogenesis process, and similarly malate can be converted into pyruvate as well with the help of malic enzyme, and it will give you again glucose molecule. Citrate, important molecule, and it can be converted into fatty acids and sterol if there is a need of the fatty acids in our body. Means if the ATP levels are high and still there are pretty much process glucose levels are available, citric acid cycle is continued, then ultimately that citrate level will be converted into fatty acids, and that could be utilized in certain fertility hormones production. Then comes the succinyl CoA. Succinyl CoA can be converted when there is a need, can be converted into porphyrin, and heme. We know that heme is an important molecule of hemoglobins which help in carrying oxygen to the tissue. So that was all about the TCA cycle or citric acid cycle. Don't forget to subscribe my channel. See you in the next lecture. Bye bye.